with our very first slide, which is just a quick, brief interview into, or revision if you want, of where DNA can be found within a cell. So as you would have learned in grade 10, the site where we will extract DNA from, that will be inside our nucleus. However, when we look at other places where it could be found, we can also find it in our mitochondria, which is the structure here, as well as the chloroplasts, which are found in plant cells. So this is probably one that you might not have learned about in grade 10, but definitely a place where we can isolate it from, that you should know of for grade 12 level. Right, so in terms of understanding the difference between a gene and a chromosome and a chromatin network, I always tell my learners to go and learn the terminology behind it. The first thing that we need to understand is that DNA is a double helix and it is made up of genes. And you would have probably learned that it consists of A's, T's, C's, and G's. So that's like binary code for computers where you have ones and zeros. Our binary code is A's, T's, C's, and G's, and they code for particular genes. So that can be found on the DNA strand. So when we have a look at our image on our PowerPoint presentation, we're going to start with that DNA double helix. So here we can see it's like a twisted ladder, and a portion of these bars, which contain the nitrogenous bases, that will be a particular gene that we might be looking at. So that codes for a characteristic like eye color or hair color. Now, you can only imagine how many characteristics there would be coding for a human being. So we've got this super long stretch of DNA, which we need to fit into a tiny nucleus, and obviously that would be a challenge. So what the cell has done is it would take the DNA and it would wrap itself over and over around these protein molecules, which we call histones. So eventually it's super coiled into a structure which we know as a chromosome. So right here at the end, that is then the super coiled structure. So if I had to unravel my DNA, eventually right at the end, I would see it as a double helix. So it pretty much comes down to your DNA is found inside the chromatin network, and depending on what the cell is doing, whether it's prepping to divide or if it's already going into division, like cell division, then you're either going to see it as a chromosome, or if the cell's just performing its normal function, that DNA will be found inside the chromatin network. So we're going to quickly go through our slide in terms of understanding the concepts. Chromosomes are thread-like structures found in the nucleus. They're composed, its monomer is of DNA. It's wrapped around protein structures, which we call histones, which are these yellow balls that we see in the picture. And that pretty much makes up our DNA strand. Okay, in terms of the history of DNA, these three characteristics or characters that we see in front of us will be very important for your exam. They can ask you a question like, who is responsible for discovering the structure of DNA, which most of you will know as Watson and Crick, these two guys here on either side of the double helix. So they're the ones that designed the structure in terms that it's a twisted ladder, consists of A's equal to the amount of T's, C's equal to the amount of G's. But their understanding of the structure was actually helped along by Rosalind Franklin. So Rosalind Franklin was also working on the structure of the DNA, and she actually took a photograph of what the DNA structure would look like. So it's an X-ray photograph, which her assistant, Maurice Wilkins, actually showed to Crick, and from there they were able to come up with the actual DNA molecule structure. So they went down in history for the discovery. Unfortunately for Rosalind Franklin, she already passed away by the time the Nobel Prize was awarded. So Watson and Crick, along with Wilkins, received a Nobel Prize for that discovery. Right, so we're going to move on to our next slide.
All right, so looking at the structure of DNA, what we have here are two backbones made up of a sugar and a phosphate. So what you'll see here is a deoxyribose sugar as well as a phosphate, and they make up your backbone. So you'll see over here we have a sugar, phosphate, and then on the other side of our helix we also have a sugar, phosphate. So this make up our backbone, as I mentioned, and it's quite important that you know that sugar, in terms of DNA, is referred to as deoxyribose. All right. That is quite different to the sugar found in RNA, which we'll get to in a little bit, but the sugar in DNA is called deoxyribose. Then the monomers of DNA, we refer to these monomers as nucleotides. So there, just again on the slide, we have the phosphate groups, then we have the deoxyribose sugar, and then we find our nucleotides. We'll take a little look at these nucleotides from like a zoomed in perspective, but before that we also have these nitrogenous bases that we find in the middle of our backbone. So Yolandi mentioned the A's and the T's, the C's and the G's. So those are our nitrogenous bases, and one nitrogenous base with a deoxyribose sugar with a phosphate, that makes up our nucleotide. So looking at this nucleotide, we have a sugar molecule, which I mentioned, a phosphate group, and then a nitrogenous base. For illustration purposes, the sugar is always represented by this pentagon structure, and then the sugar has then two things attached to it, a phosphate on the one end and then a nitrogenous base on the other end. There are 4 nitrogenous bases. Those are then our A's, T's, C's and G's. The A stands for adenine, T for thymine, C for cytosine, and the G for guanine. And then quite an important concept is that they are extremely matchy-matchy. The A and T nitrogenous bases, they are complementary, so they will only match up with one another. And then the G's and the C's, the same thing, they are also complementary. So that is an important concept, which is important for the whole process of protein synthesis and some of the questions that you need to be able to answer. So looking at this strand, we have a single strand of DNA on this side. The reason why we know this is DNA is because there's a thymine. If you have done this chapter before, you know that thymine will be replaced by a uracil when we have RNA. So looking at the complementary DNA strand, that would then, the complementary nitrogenous base over here would be thymine, which is complementary to the adenine. The guanine will be complementary to cytosine, etc. So just looking at this diagram, you need to be able to give me a few labels. So we can ask you to identify that orange circle. So that orange circle is called a phosphate or a phosphate group. Then that blue pentagon is a deoxyribose sugar. You can't just give the label just a sugar. Right, it is a sugar, but be specific. The type of sugar it is a deoxyribose sugar. Then between our nitrogenous bases, between the A's and T's, C's and G's, we have a hydrogen bond. More specifically, a weak hydrogen bond, because that bond will need to break at a later stage with the processes involved in DNA replication as well as with transcription. Alright, so looking at our complementary bases, the complementary base for the next one, G, will be C, and then I'm pretty sure that you can complete the rest. One last concept that I want to just clarify is that the amount of adenine bases and thymine bases, they will always be equal. So if you add the amount of adenine bases, we have 1, 2, 3, 4 bases of adenine, then there needs to be 4 thymine bases as well. Right, so since they are complementary, we have equal numbers of adenine and thymine bases and equal numbers of cytosine and guanine bases.
Just want to quickly, while we're on this slide, there's a cute mnemonic that my learners make use of. They call it apple in the tree and car in the garage. So the A for apple goes with tree, T for tree, and then car in the garage, C and G go together. And if you have these mnemonics that you can just like quickly set up for yourself before a test, and it really helps with memorizing it, and especially when it comes to the DNA structure. So we're going to look at DNA replication in a short while, but when we talk about protein synthesis, then things heat up just ever so slightly. Right, so we're going to look at our next slide, which is on DNA replication. And for this process, we need to understand what is DNA replication. And that's pretty much just taking this piece of DNA, and we are literally just making more genetic information in preparation for a cell to separate. So we will have another lesson soon on meiosis, and then this all fits in with that section on when we are splitting a cell. We don't want to lose genetic information, so we have to first undergo a process of DNA replication so that we can have a double-stranded chromosome. Okay, so when does it take place? This always takes place during interphase, and then where does this take place? This occurs within the nucleus. So just to quickly put this into perspective, if I have a look at the page here in front of me, I've quickly drawn our chromatin network. So this we understand that this is inside of the nucleus. And if I want to divide a cell, obviously there are now all these little individual strands of genetic information. So if I split this cell through the middle, this nucleus as well, then obviously the amount of genetic information that will go to left and to the right hand side will no longer be equal. So in order for us to divide our cell equally so we don't lose genetic information, these strands shorten and condense to form unreplicated chromosomes. And then through the process of DNA replication, we now make another strand of genetic information. So this chromosome then consists of 2 chromatid legs attached to the middle at the centromere. And the genetic information that we find here will be exactly the same as the genetic information that we find on the other chromatid leg. And the same applies for this piece of strand there, same genetic information to what we find here. So if the cell is going through the process of meiosis, we can now split this guy through the middle, and the 2 chromatid legs can separate at the centromere, and the 2 cells will now contain the exact same genetic information. So it's thanks to interphase, okay, which is just before prophase, metaphase, anaphase, and telophase of meiosis and mitosis, that we have this important step taking place in order to make sure that our genetic information is not lost. So let's quickly look at the practical application of the process of copying the genetic information that we have. So this is a step-by-step process, and the best way to do this is to learn it like a parrot. Repeat, repeat, repeat. So write it over and over and over until you memorize this section really well. Okay, so we're going to start with step 1, and as we can see here, this is where our DNA double helix unwinds. So literally goes, and instead of it being a twisted ladder, it now takes on a more ladder-like appearance to it. Step 2 will be for those weak hydrogen bonds that keep the 2 halves together to break, and it almost looks like a zip that is opening up. So we have a zipper, which is represented by an enzyme, and that enzyme is running down all these nitrogenous bases, breaking those weak hydrogen bonds in between them. So eventually we now have, there we can see our little zipper going, that's the little enzyme molecule. Eventually we have these 2 separate strands now that will form as a result of that enzyme action. Right, so now both these two strands will act as our templates. And I always tell my learners, when you go into a Word document and they ask you, what type of document would you like to open? And they've got all these template strands there or template documents there. And you type on, I want to make the one that says CV. Okay, that's a template. So you already have a basic outline as to what should go in there. Now, when the DNA molecule unwinds, the two halves will serve as those, templates that you would work off of. So if the one side has got TAC, then obviously what needs to go onto it will be AT. I can't remember. What was that one? See. So in terms of the binding, if we go to our PowerPoint presentation, we need to make sure that the complementary DNA nucleotides that come in and bind onto the template strand has to be complementary. So for instance, there we can see the template had a T on it, so the DNA nucleotide that has to come in has to be an A. Here we've got an A, so the nucleotide that comes in has to be a T. There we've got a C, so then the nucleotide has to be a G. So eventually we build this complementary strand onto our DNA, and we actually have to indicate how they bind. So in a test or exam, always remember to write it out completely. Adenine A binds to T, which is thymine. Guanine, which is the G, binds to cytosine, which is the C. All right. So let's say, for instance, I had to ask you in a test, what does this letter A represent? Then you would need to know that it's adenine. Okay, eventually what we will find is that there will now be two new strands of DNA, and they will be identical to each other in all ways and forms. Okay, because originally these two halves, they used to fit on top of each other, and we are literally just making a replica of this molecule. Right, so why is DNA replication so important? So as we mentioned earlier, we want to form the double the genetic material so that when the cell goes through division, that we have the same amount of genetic information present. So that's where we looked at the replicated chromosome consisting of two chromatids. So when it splits during meiosis, that we still have the same genes present in both the new gametes that form. Okay, then it results in the formation of identical daughter cells during mitosis, which links in with what we have learned during our grade 10 year. Right. So then we can also see that DNA, obviously being very unique to individual, if I had to go and look at all my skin cells, the exact same genetic information will be present in it. Now, forensic scientists can make use of that in order to determine if I was perhaps at a crime scene. They can determine maternity and paternity. So our genetic information gives a lot away about who we are. So through the process of gel electrophoresis, they can set up a DNA profile, which pretty much is your DNA bands that have separated based on size. Now, we call the process of making such a profile DNA profiling. So through the machine, gel electrophoresis, we make a DNA profile, and the scientists can then sit, or the forensic analysts can then sit, and they can solve a crime with it. So when we quickly have a look at such an application question, because typically it will be asked as an application question in test or exam, they will sketch a scenario. For instance, in this one that we see here alongside, it says that there was blood taken from broken glass. So this is an unknown individual's blood sample. And then they will have either suspects or victims or whatever the case might be, which they've labeled here as P, Q, and R. Now, that person's DNA profile will be unique to him. So all these DNA fragments that we see here would need to match 100% with the victim's DNA profile. So we're going to look at the size of these DNA fragments, and we're also going to look at the position of where these guys are found in order to determine who the person, or where that DNA, who does it belong to. So the first question is, what is DNA profiling? And it's pretty much a summary, a pattern of black bars, and these black bars represent a person's DNA fragments. And the uses of it is either for paternity testing, we can use it to trace missing individuals, we can look at identifying genetic disorders, or we can use it in forensic investigations, as we see in this scenario that we've got alongside. So let's say that we do see this question in an exam, and the question states, when a thief broke into a car, he cut his arm on the broken glass. Scientists extracted the DNA from the blood found in the broken glass, because remember, things like blood and saliva all contains your DNA. They analyzed this DNA sample and compared it to the DNA from 3 suspects. So we've got P, Q, and R. The table below shows the results of the analysis for the DNA from each individual. So for this forensic component that we have here, we have to look that all the bands in the DNA sample need to match 100% with that of the individual in question. So I normally tell my learners, take a ruler and then put a ruler underneath the bars, make sure that they are the same width, and then you will be able to tick off next to the bars if you see a match. So already I've got 1 tick here for this individual. If I go along, I see these guys are also in the same position as well as that one, so I can put a tick next to it. Then I see here's another bar, and it links up with individual Q. Already it looks a little bit suspicious. Okay, then we've got another one here that matches with person Q yet again. Already we're almost here on 100%. There we've got another match again as well. This guy's also matching. That one's a little bit further up. And then our last one makes it a 100% complete match with individual Q. So that can then be said without a doubt that that must have been the person that broke the glass and that was involved in the crime scene. Okay, so examples of DNA profiling can be a crime scene. We can also have a situation where we need to determine paternity testing. Let's say, for instance, that there's a baby and the mother's not entirely sure who the baby daddy is. There's a lover and there's a husband. Then we need to take the DNA profile from the baby, from the mother, from the lover, and the husband in order to go through a process of elimination. So we're going to look at the mother's contribution towards the child, and then from the dad's contribution to the child to do this process of elimination. So in terms of what we see, this whole DNA fragment here was given from the mother to the child. This fragment here was given from the mother to the child. Here we see another fragment passed down from the mother. So the remaining fragments in the baby had to have come from the husband. So in this case, the husband was definitely the father of this child, and the lover obviously did not feature. Right.
Okay, so moving on to the second type of nucleic acid, we have RNA. And as mentioned, RNA also has nucleotides. But in this case, there are just a few slight differences in the nucleotides of RNA in comparison to the nucleotides of DNA. So RNA still has that sugar molecule, but in this case, the sugar molecule is ribose instead of the sugar molecule being deoxyribose. So there's sugar, ribose sugar. We still have a phosphate group, and then we also still have a nitrogenous base. So looking at our slide, you need to be able to give labels to these parts. So that orange circle over there would be the phosphate. That pentagon structure again will be a sugar. And because I'm telling you that it is RNA, that sugar is called a ribose. And then again attached to the sugar, we have nitrogenous bases. So hopefully you can figure out that this nitrogenous base over there is a bit different to the nitrogenous base found in DNA. So that nitrogenous base is called uracil, and uracil is exclusively found in RNA. Uracil technically just replaces thymine, which is found in DNA. Right, so RNA is a single strand. That's another difference of RNA compared to DNA, which was a double strand. And then, as I mentioned, this nucleotide again is made up of a sugar, a phosphate, and a nitrogenous base. This nucleotide is made up of a ribose sugar, phosphate group, and then nitrogenous base. And these 4 nitrogenous bases are adenine, cytosine, uracil, which is one way of figuring out whether we're dealing with DNA or RNA. When you see uracil, we know that we are referring to RNA and not DNA. And then, of course, guanine. So again, in terms of complementary base pairing, adenine will be matched up with uracil, and then cytosine and guanine, again, they will be complementary. There are 2 types of mRNA that we are quickly just going to mention, and they play a role in the whole process of protein synthesis. So mRNA is called messenger RNA, and this literally just takes a message from the DNA. Because DNA can't move out of the nucleus, we need sort of a copying system, something that takes a message from the DNA and will be able to move out of the nucleus to now make something out of the instructions found in the DNA. So mRNA is a messenger RNA. It is made in the nucleus, and then it will move out of the nucleus. Right. So this picture shows us, you might have seen this from the, what is that document called? Mind the Gap. So over there we have our DNA. You can see it's a double helix. And then Yolandi will speak to us about how this mRNA is made. But that little single strand made from the DNA is then called mRNA. And there it shows you an arrow. It will move out of the nucleus and attach to a ribosome at a later stage. So it is basically made during the process of transcription, which we will tackle in a little bit. And it's formed in this nucleus, and then later on it will move to the ribosome. So you need to be able to identify, they refer to this as a molecule. Right, so you need to be able to identify this molecule, and then you can say with confidence this is mRNA. The second type of RNA is called tRNA, and that T stands for transfer, because it will transfer an amino acid. Right. So on the one side of this entire molecule is called a tRNA, and that little polka dot there at the end is an amino acid. And basically, during the process of protein synthesis, we are going to link many amino acids together to form a protein. So this transfer RNA will now bring an amino acid, it will transfer it to the ribosome to eventually make a protein. So that is just a little introduction about our 2 tRNA molecules, mRNA and transfer RNA. So now back to the big business, protein synthesis. So now we'll look at how these 2 molecules, what their part is and what their role is in this major process. So the 2 processes are called transcription. It takes place in the nucleus. And the second process is called translation, and that takes place outside of the nucleus in the cytoplasm. In terms of these 2 processes, you need to be able to give a good description of both of them. So that is easily found in your exam guidelines, where you can just follow the basically the steps involved. And then you also need to be able to answer basic questions that is depicted in a diagram. So what we'll do next is we'll discuss what happens during transcription, what happens during translation, and then I think we'll still have some time to look at a few questions about protein synthesis.
Okay, so I'm going to stick to this picture that we have in front of us and just quickly go over transcription and translation, because I know that a lot of students struggle with the concept of when does transcription take place, when does translation come place, which one is first, which one is second. And just a simple way that I think about this is the word scribe, transcription, scribing. So I make that association. And the question is, what are we scribing? We are scribing a coded message. So your DNA, all those A's, T's, C's, and G's is like a foreign language. You've never been able to decipher what those A's, T's, C's, and G's mean. But think about the big picture. Your DNA is an instruction manual on how to make a protein, okay? And what are we trying to make here? We're trying to make a protein which is either like hormones or enzymes. So we have to do a little bit of revision. And the first thing is that the nucleus contains the genetic information, the instruction manual for the cell on how to make a protein. And the site where the protein will be made is obviously the ribosome. So that's the site of protein synthesis, which you've learned in grade 10. So that's a connection between the two. But the problem is that your DNA cannot leave the nucleus. It has to stay inside of it. So I think about this like the principal sitting in her office, and she's the one that tells the teachers what subject they must teach. And then the students are sitting there in the front of the class and then passing the exams. That to me is almost like the protein that's being made. So the principal, she's the head, she knows the bigger picture, she assigns to the teachers like what they're meant to do, and then the students in the class, they're like the product that is formed at the end of the day. Now, there are different ways or different analogies that you can use. For instance, you can also think of the DNA as being this foreign recipe that's in a language that you don't understand. So let's say that you're on this foreign country, you see this beautiful cake, you want to make this cake, the cake represents the protein, just so FYI. And you go to the library, you visit it, you can't take the book out, so what do? You write down the recipe on a piece of paper. Now what do? You take that coded message, that foreign language, A's, T's, C's, and G's, and you find yourself a translator. So you go to a chef, the chef reads this coded message, you are now suddenly this tRNA molecule running around getting ingredients, and the chef makes you a beautiful cake. So this all is the translation component to it. So what it comes down to is transcription has to happen before translation can occur. All right, so we're going to dig into this process, starting with our very first part, which is transcription. And the word transcription, if we look at it, we also have the letter C in it, and C comes before L. So that's another way that you can remember which one is first in the process. So our step one would be very similar to DNA replication. However, there will be a few slight differences here, in that we want this DNA double helix to unwind. So in both cases, we have to open up the structure. We have an enzyme that's going to break the weak hydrogen bonds, and then eventually we will have two separate strands. So here we have the two separate strands that have now been exposed. Those weak hydrogen bonds have been broken. Okay, so the next step that we have is that one of these strands will act as a template strand. So onto the template, we will have an mRNA molecule that will be formed, and we're going to use free-floating RNA nucleotides that are within the nucleus. So one at a time, they're going to come in, and they're going to base pair with the DNA strand. So for instance, if we've got thymine on the thymine on the DNA strand, then adenine will bind on the mRNA strand. If we've got adenine on the DNA strand, then uracil, which is a new one, is now going to bind in its place. Cytosine will still bind to guanine, guanine will still bind to cytosine. But on the RNA strand, the only difference will be that we no longer have thymine, and uracil will be the replacement nucleotide. Okay, so eventually we will have a fully formed mRNA strand, and that will now contain that coded message of use A's, C's, and G's that can be used to synthesize the protein molecule. So that's now going to move through the nucleus, through the nuclear pore, and the DNA will be allowed to obviously form a double helix again. Right, so here we can see this guy's now going to make its way out. Okay, there we've got our mRNA strand that was formed. This is now a zoomed in version of the process of transcription that we just ran through, where we can see that it only consists of U's, C's, A's, and G's. There are no longer thymine present. And just like with any code, we need to decipher it. So on our mRNA strand, the code will be read 3 nitrogenous bases at a time. So if it's on an mRNA strand, if we look at 3 of them at a time, we refer to those 3 nitrogenous bases as the codon. So it's a triplet because it's made up of 3 nitrogenous bases. So in this strand that we have here on the screen, we've got 1 codon, a 2nd codon, and a 3rd codon being represented. So they might ask you how many codons there are. They might ask you how many nitrogenous bases do you see. So then you would have to count all the individual bars. And you might be asked to answer more complex questions. We have to link the codons to amino acids or to tRNA structures as well. Right, so just to recap, this all takes place inside of the nucleus. And we want to form an mRNA strand off of the DNA. We only use 1/2 of the DNA molecule, which acts as as the template, and then that will be allowed to leave the nucleus.
All right. So looking at there's just a little bit of a crossover between transcription now and translation. To have a nice little representation, here we have a DNA template strand. So again, that DNA molecule is going to unwind, the hydrogen bonds will break, and we will basically have 2 separate strands, and only 1 strand will act as a template. So if you look at this picture, there are T's involved. Okay, so that gives it away that we are referring to DNA, not RNA yet. And again, just like Yolandi said, we read them in 3 nitrogenous bases at a time. On a DNA strand, the 3 bases is called a base triplet. So there'll be an enzyme that will basically make an mRNA, this copy. It will transcribe the message from the DNA. And over here, you can sort of see the complementary base pairs. If there was a T on the DNA strand, the complementary nitrogenous base would be adenine. If there was an A on the DNA strand, there'll be a uracil on the RNA strand. Right, so this molecule, this red one, this will be known as the mRNA molecule. And this mRNA molecule, again, we read them 3 nitrogenous bases at a time. Those 3 will then be codons. So this mRNA molecule now has the ability to leave the nucleus. The poor DNA has to stay behind. As it leaves, it is going to now attach itself to a ribosome. So the mRNA will go to the nucleus, out of the nucleus, attach to a ribosome, and now we are ready for the 2nd stage, known as translation. So this is still, it says transcription, but this is technically the start of translation. Right, so there we have our ribosome, and coming to the ribosome, there'll be a transfer RNA molecule. So tRNA will basically be floating around in the cytoplasm, and it will contain these another three nitrogenous bases on the one end, and those 3 nitrogenous bases are referred to as an anticodon. Right, and on the other side of this tRNA, there will be an amino acid. So if you can remember, I mentioned earlier that the transfer RNA will carry an amino acid on the one end, and then it will also basically have this 3 nitrogenous bases on the other end. And those 3 nitrogenous bases are referred to as anticodons. So now we have to play the matchy-matchy game again, and these 3, the anticodon, need to be complementary to the codon on the mRNA. So if you read here, the mRNA codon reads AUG, so the complementary tRNA, the anticodon, will then read UAC. Right, and that then brings a very specific amino acid to the ribosome. Okay, so this transfer RNA, it brings in an amino acid, and it will basically match up to the codon, which will then be complementary to the anticodon. Okay, so in terms of labels, you need to be able to give labels to that structure over there, which will then be carried by the tRNA, the anticodon, and that structure up there, that would then represent the amino acid. Okay, so after that tRNA has matched, the anticodon has matched with the codon, there'll be another tRNA that will bring another amino acid to our ribosome. Okay, so here comes another tRNA molecule bringing about a nice pink amino acid. And if you see look at the next codon, it reads GGC. Then the complementary anticodon would read CCG. Okay, so the next second tRNA is sort of brought to the ribosome, and then something special needs to happen. These two amino acids, they need to link up because we want a whole chain of amino acids. Because we want to make a protein and we need a whole chain of them, so they can't just be loose next to each other, they have to bind. So there's a special bond known as a peptide bond that will form between them. Alright, so there we have that peptide bond, also a term that you have learned about in grade 10. And then we have bonded those two amino acids. The first tRNA is now sort of relieved, so it can go, and another tRNA molecule can now actually be brought to the ribosome. So then the whole system sort of repeats. Another tRNA molecule will bring another amino acid. In this case, we have a yellow one. If you look at the sequence, it reads UUA on the codon on the mRNA. So the complementary anticodon now has to read AAU. And then again, that anticodon brings a specific amino acid to the ribosome. So it will transfer that ribosome to that amino acid to my ribosome. Okay, so here's another fast little interpretation of another tRNA molecule binding the anticodon and the codon binding together, and basically we see that link, that peptide bond that will form between my amino acids. Okay, so again, I need another peptide bond to form between my amino acids. We can't just have loose amino acids hanging around. They need to be bonded together, again by a peptide bond. So then eventually we have a polypeptide chain, which will then, when we have a long chain of amino acids, it will form an protein.
So that's usually about 50 or more amino acids that have been strung together.
Yes, this is a very big process. You need to be able to give a whole description of the process of transcription, the process of translation, and that basically that description of the processes is found in your exam guidelines, so you can find that and memorize them. And then in terms of deciphering these images, usually they give you a little bit of a clue and tell you this is a ribosome or this is an mRNA, and then you need to sort of orientate yourself and figure out where's this process taking place. Find little clues to help you to just help you to figure out what is taking place.
So typically what they can do in the exam, like Monica has just mentioned, your examination guideline will contain an outline of these two processes, transcription and translation. And the best advice would be to learn it like a parrot, okay, word for word. If you can write it out, I would say if you can write something out as an essay, you would be able to apply it, okay. So we also need to understand what is actually happening within this process of protein synthesis. So normally what I do is I take a blank sheet of just the images, the nucleus and the DNA, and we see the mRNA that goes out of the nucleus, and there's a picture of the ribosome there, and we've got our little amino acids floating around in the tRNAs. And then I look at this diagram of protein synthesis and I repeat it over and over and over in my head. Okay, and then this happens, and then that happens, and then this happens. So that by the end of the day, if they ask you that diagram, that you will be able to answer the different questions, the application style questions that could come from it. So that's one way. They can ask you about 8 marks where you need to write a mini essay on transcription or translation, or they can ask you the application style questions. So we're going to dig into a few questions. These would have appeared in past papers, but we also encourage you to use the Mind the Gap, which contains those blank template images of protein synthesis as a study tool as well. Right, so let's go into our question section. Before we go, here's just a quick little revision on protein synthesis. So there we can see our tRNAs coming in. Those green structures actually do look like T's if you think about it. So it makes it easier to memorize it as well. So there we've got our mRNA that's synthesized by the enzyme, goes to ribosome, tRNAs come in, peptide bond forms. Eventually we've got about 50 of them strung together, and then we've got our hormone and our protein. Okay, so before we start, just a little something in terms of the amino acids that are formed. Remember that you only have 20 different types of amino acids, and depending on how they are strung together will depend on what hormone or enzyme has already been formed or is being formed. So for every species, we actually look at this utilization of amino acids and making proteins to also see how related they actually are to each other. So just a little something extra that you might have read up somewhere. Okay, so let's jump into our first question, and here's that image that I've already shown you guys in I spoke about from Mind the Gap. So this is the actual image that you can sit with in front of you and just repeat and talk over. So for the very first question that we have, they ask us, what is molecule A? So here we've got the structure. We can see that it's that double helical DNA molecule. Ooh, they give us all the answers all at once. So let's just quickly go through it step by step. So here we've got our DNA double helix. I always tell my learners, it's a good idea just to write it all out, but it is the DNA. Then we need to find molecule B, and that represents our mRNA strand. We can see that it is single-stranded, which is also one of the requirements for RNA, while DNA is double-stranded. So that's how we identify it. Okay, then we're going to look for C, which is part of our nuclear membrane, so the opening that our mRNA molecule can just slip through. That will be our nuclear pore. And then structure D, the whole structure that we see here, this will then represent our tRNA. So there we go. There's the answers to that structure. Take note of how they have written down mRNA. It's a small letter for the M, and then all capital letters for your RNA. And the same applies to your tRNA and how you write it out as well. Okay, so now they ask us for 1.1.2 to tabulate two differences between the monomers of molecules A and molecules B. So here we've got A, which is our DNA. Here we've got B, which is our mRNA. And they want to know about the building blocks of these two structures. So we know that nucleotides make up these two structures here. And one of the big differences that we find in DNA is that obviously the nitrogenous bases will be T, A, C, and G, but the nitrogenous bases for our mRNA molecule will be U, A, C, and G. Right, so when we go through that, okay, here they mention about the nitrogenous bases. Thymine is found as a nitrogenous base in DNA. Uracil is found as a nitrogenous base in mRNA. There's no longer thymine that's present. But we also have learned about the sugar component that makes DNA. So the best way to look at this is think, what does DNA actually stand for? And it stands for deoxyribonucleic acid, while your RNA stands for ribonucleic acid. And the name is derived from the sugar that is present inside that nucleotide structure. So in the case of DNA, it's deoxyribose, and in the case of RNA, the sugar component is just ribose. So very important to memorize that one, that if they ask you a nucleotide, you can't just label it sugar, you have to be specific. Is it a deoxyribose sugar, or is it a ribose sugar that's being assessed? Right, so then we get to question 1.1.3, and they ask us to describe the process of translation during protein synthesis. Okay, so now we're in the second half of our process of protein synthesis. So we're looking at how do we actually take this coded message that we have here and make a protein molecule from it. So we're going to start off with the first step, which is that a tRNA transfer. Remember, its job is to transfer something. What is it transferring? It's taking amino acids which are floating around in the cytoplasm, and it's picking them up. So the anticodon on the tRNA will be specific for a particular amino acid. So now they go and find the amino acids, they go and pick it up, then they go and look for the codon on the mRNA that's complementary, anticodon to codon. So as soon as it has found that, it will bring it to the ribosome, attach anticodon to codon. Now the amino acid is in place. So we are waiting for a second tRNA to come in, also with its amino acid attached to it, waits for codon and anticodon to bind together, and that will then allow for the two amino acids to have a peptide bond that forms in between them. So as soon as we have another one comes in, same story, anticodon to codon matching. Eventually, if we've got about 50 or more amino acids strung together, we finally have our protein molecule. Anything less than 50, that will be considered a polypeptide molecule. Right, so that is the process of translation then. So then they can start to heat it up slightly. And they say that we've got a section of DNA molecule that has the following base sequence. So we've got C, T, T, A, C, A. And they want to know the percentage of guanine in this DNA molecule. Okay, so they are telling us that there's about 30% of it. Now they want to know, give the percentage of thymine in the same molecule. So I always say to my learners, if they give you a question like this, to write it down. Okay, this is like a mathematical equation, and in this case they've got guanine, and it always binds with cytosine. So if we've got 30% of it being guanine, there will be 30% of the molecule consisting of cytosine. Okay, as you will recall, Monica explained earlier that your G's will be equal to your C's. Then we also have T binding to A. So now that's just the leftover amount. So if we are working out of 100%, we deduct that 60 together from the 100%. That leaves 40 left over. So we have to split that to the remaining amount, which will be 20% of it being T's and 20% of it being A's. So if we look at our answer for this one, to answer it, that will be 20% thymine. Okay, so unfortunately, that is all that we have for today. You will be able to find the rest of the questions on our telematics website, and the memo for all the questions are present there as well. So yeah, we encourage you guys to go work through some examples and just look as many past papers as possible for this section. So I hope you enjoyed our lesson today. Until next time.
Bye.